Hello everyone, this is Charmaine Espinosa, and thank you for your interest in this podcast. Today, we will talk about a single micronutrient that is commonly included in diets in minute amounts but exerts great impact on performance and health in pigs, which is copper. Copper, as we all know, has been recognized as an important micronutrient needed for maintenance, growth, and optimum health. And that is because copper is involved in several metabolic reactions, including cellular respiration, tissue pigmentation, and hemoglobin formation. Copper is also an essential component of several metalloenzymes, including cytochrome oxidase and lysyl oxidase, and is involved in oxidation reduction reactions and protection against oxidative stress. The requirement for copper by weanling pigs is 5 to 6 milligrams per kilogram in the diet, and if this amount is included in diets, deficiency symptoms are not observed, and we expect that animals will be of normal health status. And this small requirement for copper is usually provided by our plant and animal-based feed ingredients, as well as the inorganic copper present in our vitamin mineral premixes. However, copper may also be included at growth-promoting levels in diets for weanling and growing pigs. Copper, included at a range of 75 to 250 milligrams per kilogram in diets, usually exerts positive effects in pigs, and as early as in the 1950s, it was demonstrated that high levels of copper increased growth rate and feed efficiency in pigs. We actually determined the effects of pharmacological levels of copper in our pigs about 5 years ago, and here we only had 2 dietary treatments. The control diet containing approximately 20 milligrams per kilogram of copper, and the diet containing supplemental copper at 150 milligrams per kilogram. And what we have observed is that the incidence of diarrhea was reduced when pigs are fed a diet with supplemental copper, and this result has been constantly demonstrated in 2 additional experiments that we have conducted for copper. With the reduction in diarrhea incidence, that followed an improvement in growth performance, where here we can observe that supplementation of copper at 150 milligrams per kilogram increased average daily gain and feed efficiency in nursery pigs. We conducted another experiment to determine if supplemental copper also exerts positive effects on challenge pigs. So in this experiment, we had 4 treatments: a control diet, a diet with 5% fat, a diet with 100 milligrams per kilogram of copper, and a diet with 5% fat and supplemental copper. And heat stress was induced in pigs fed these diets. Obviously, gain-to-feed ratio of pigs increased with the addition of fat, as we can see here, but we can also observe here that by merely adding 100 milligrams per kilogram of copper in the diet, pigs had increased gain-to-feed ratio similarly as what we have observed in the diet with 5% fat. Therefore, the negative effect of heat stress in growth performance can be alleviated if we include high levels of copper in pig diets. With a similar effect of copper as with fat in relation with increasing feed efficiency in pigs, we hypothesized that copper may increase the energy value in our diets, and it was speculated that this is due to the ability of copper to improve animal fat utilization. To verify these speculations, a recent experiment was conducted to determine the energetic value or fat equivalency of dietary copper in comparison with choice white grease. So here in this experiment, 144 pigs were used with 2 pigs per pen and 12 replicate pens per treatment. So these pigs were randomly allotted to 6 dietary treatments, and the first diet is formulated based on corn, soybean meal, and distillers dried grains with solubles or DDGS. 3 additional diets were formulated by adding 2, 4, and 6% choice white grease to the basal corn soybean meal DDGS diet. 2 additional diets were then formulated by adding 150 milligrams per kilogram of copper to the basal diet and to the diet containing 2% fat. And these diets were fed to pigs for a month. Moving on with the results, this figure shows the gain-to-feed ratio of pigs fed diets containing increasing levels of choice white grease and 150 milligrams per kilogram of copper without or with 2% choice white grease. So the bars here are represented with increasing intensity of orange to red as inclusion of choice white grease in the diets increased. For the overall experiment, the gain-to-feed ratio of pigs linearly increased as the concentration of choice white grease increased in the diets. So, this linear improvement allowed us to conduct a linear regression analysis, and a prediction equation was derived with 0.526 as the intercept and 0.022 as the slope. This indicates that gain-to-feed ratio of pigs increases by 0.022 for each percentage unit change of choice white grease inclusion in the diet. You can also observe here that supplementation of copper improved gain-to-feed ratio of pigs from 0.526 to 0.609. And based from the prediction equation, it was calculated that the improvement in gain-to-feed ratio upon copper supplementation was similar to the improvement obtained by adding 3.8% choice white grease to the diet. Likewise, inclusion of 2% choice white grease and supplementation of copper to the diet improved the gain-to-feed ratio of pigs from 0.526 to 0.632, which resulted in a 4.8% choice white grease equivalency, 2% coming from choice white grease and 2.8% coming from supplemental copper. Therefore, what we can conclude in this experiment is that inclusion of choice white grease and supplementation of copper to diets improve gain-to-feed ratio of pigs, and the improvement in gain-to-feed ratio upon copper supplementation was similar to the improvement obtained by adding 2.8 to 3.8% choice white grease to the diets. So how does copper exert these positive effects in feed efficiency of pigs? Now we want to shed some light into these questions, and therefore, we conducted an experiment to test the hypothesis that copper reduces endogenous loss of fat and also improves the digestibility of fat in these pigs. In this experiment, 64 pigs were used and were allotted to 8 dietary treatments for a total of 8 replicate pigs per diet. A basal diet based on corn, soybean meal, and corn bran at 27% was formulated. 3 additional diets were then formulated by adding 15, 30, and 45% DDGS to the basal diet. Now I want to emphasize here that as we increase the inclusion of DDGS, the dietary concentration of fat also increased. And you can also observe here that the concentration of corn bran was reduced as DDGS was increased to maintain a constant concentration of dietary fiber among these diets. Lastly, 4 additional diets were formulated by adding 150 milligrams per kilogram of copper to the first 4 diets. These graphs shows the apparent all-track digestibility of fat in diets with increasing concentration of DDGS without and with supplemental copper. Blue bars here represent the digestibility of fat in diets without added copper, and the orange bars here represent diets with copper. We can observe the digestibility of fat linearly increased as the concentration of DDGS increased in diets without or with copper, which is likely a result of the increased concentration of dietary fat in diets. What is also interesting here is that supplementation of copper to the diet without DDGS and to the diet containing 15% DDGS improved the digestibility of fat. The same is the case here, wherein supplementation of copper to the diet containing 30% DDGS tended to improve fat digestibility. However, if we take a look into this graph showing the apparent all-track digestibility of gross energy, copper supplementation did not improve the digestibility of gross energy which is in contrast to the observed improvement in fat digestibility upon copper supplementation. Well, this was due to the effect of copper on reducing the endogenous loss of fat from 11.2 to 7.1 gram per kilogram dry matter intake. We also estimated the true total tract digestibility of fat of pigs fed diets without or with supplemental copper, but we did not see any difference. Therefore, the improvement in the ATTD of fat upon copper supplementation is only due to the reduction in endogenous loss of fat. With the result that we have observed from previous experiment, it is hypothesized that this is likely due to the ability of copper to modulate microbial activities in the intestinal tract of pigs. Dietary copper reduces the number of bacterial species in the small intestine and in the cecum and colon of pigs, and this may have reduced fermentation of fiber in the hindgut with a subsequent reduction in endogenous loss of fat, since majority of fecal fat is of microbial origin. Copper may disrupt enzyme structures and functions of bacteria, and indeed it was demonstrated in a previous experiment that a high copper diet did not improve growth performance of germ-free pigs, but the high copper diet increased the average daily gain in conventionally reared pigs, indicating that copper can alleviate the negative effect of pathogenic microorganisms in pig performance. To indirectly confirm if copper changes microbial activity in the intestinal tract, an experiment was designed to test the hypothesis that supplemental copper changes the concentration of microbial protein in the large intestine of pigs. Here we have 4 treatments in a 2x2 factorial arrangement, 2 levels of DDGS and 2 levels of copper at 0 or 150 milligrams per kilogram. And if we take a look into this graph, fecal microbial protein concentration increased if diets contain DDGS, whereas copper supplementation reduced the concentration of microbial protein in feces. Increased concentration of fiber in diets upon DDGS inclusion results in increased fermentation by microbes in the intestinal tract. On the contrary, the observed reduction on fecal microbial protein concentration upon copper supplementation is likely a result of the bacteriostatic properties of dietary copper, by which it has the ability to disrupt enzyme structures and functions of bacteria. The observed response in microbial protein further supports the results we have observed in the concentration of volatile fatty acids, where copper supplementation tended to reduce the concentration of VFA from carbohydrate fermentation in the feces. Therefore, these observations indicate that the improved feed efficiency that was observed in pigs upon copper supplementation is likely due to the effect of copper in reducing microbial populations in the intestinal tract. These may have reduced the number of toxins and pathogenic microorganisms that could have negatively affect intestinal health, which may have reduced incidence of diarrhea and positively influence immune response in pigs. Indeed, it was demonstrated that copper supplementation increased the concentration of superoxide dismutase, and it was also observed that copper supplementation to diets also reduced the concentration of tumor necrosis factor alpha in pigs, which may be a result of the impact of copper in improving the immune status of pigs. It is possible that this effect is also due to the bacteriostatic property of copper in reducing inflammation caused by pathogens. In addition to the impact on intestinal health of pigs fed dietary copper because of reduced microbial populations, the mode of action of copper was also hypothesized to be related to systemic effects. Administration of high concentration of copper via intravenous injection improved growth performance in previous experiments, which was hypothesized to be attributed to the effect of copper in increasing the mRNA expression of certain hormones in pigs. Furthermore, copper may have some effects on the postabsorptive metabolism of lipids, since in other species it was demonstrated that inclusion of 45 milligrams per kilogram of dietary copper in diets for rabbits improve body mass gain by upregulating mRNA transcription of fatty acid transport protein and fatty acid binding protein in skeletal muscle. Supplementation of copper to diets also increased lipogenesis and fatty acid uptake in fish by upregulating mRNA transcription of fatty acid synthase and acetyl-CoA carboxylase in intestinal tissue. Therefore, it is possible that copper will exert similar effects in pigs. However, data demonstrating this are limited. Therefore, we conducted an experiment to test the hypothesis that supplementation of copper at 150 milligrams per kilogram to diets improves growth performance by upregulating the mRNA transcription of genes involved in postabsorptive metabolism of lipids. 32 pigs were randomly allotted to 2 experimental diets. The first diet is formulated based on corn, soybean meal, and DDGS, and the second diet was formulated as the first diet, with the exception of adding 150 milligrams per kilogram of copper. On the last day of the experiment, we collected tissue samples to analyze for mRNA abundance of genes involved in lipid metabolism. This graph shows the mRNA abundance of genes in the subcutaneous adipose tissue. And here we can observe that the expression of lipoprotein lipase and fatty acid binding protein 4 increased when copper was added to the diet. The observed increase in the expression of lipoprotein lipase upon copper supplementation is possibly due to the role of copper as a cofactor of the activator complex of lipoprotein lipase, where the activation of lipoprotein lipase is influenced by apolipoprotein and divalent cations. These then indicates an increased uptake and utilization of fatty acids from the hydrolysis of the triacylglycerol component of chylomicrons, which is possibly the reason of the increased expression of fatty acid binding protein 4 upon copper supplementation. Supplementation of copper also increased the mRNA abundance of CPT1, indicating an increased synthesis of ATP due to enhanced oxidation of fatty acids. We know that supplementation of copper to diets for weanling and young growing pigs has been a common practice due to its consistent improvement in growth performance. However, this may not be the case with finishing pigs, as the response in growth performance is quite variable, and it is still debatable whether copper supplementation has additional benefits in finishing pigs. As an example, here in this experiment, it was demonstrated that finishing pigs had increased average daily gain when fed diet supplemented with 175 milligrams per kilogram of copper. However, Carpenter and colleagues reported no positive effect of supplemental copper at 70 or 130 milligrams per kilogram on performance of finishing pigs. However, in a recent experiment conducted this year, it was observed that average daily gain of finishing pigs increased when diets are supplemented with low levels of copper, in this case at 5 to 20 milligrams per kilogram of copper. These inconsistent responses could probably be related to the maturity of the animal, diet composition, source and inclusion level of supplemental copper, and therefore we possibly need to look at these factors that could influence sufficiency of copper in finishing pigs. Along with these factors, the optimum amount and duration of feeding supplemental copper in diets fed to pigs also needs to be further investigated. In summary, results of previous experiments confirmed that high levels of copper consistently improves growth performance and reduces diarrhea in young growing pigs. It was also a major progress for us to determine that copper supplementation improves ATTD of fat, which is mainly due to its effect on reducing the endogenous loss of fat. Because of this progress, we have demonstrated that the consistent improvement in feed deficiency upon copper supplementation to diets is likely a result of the ability of dietary copper to modulate intestinal microbial activity due to its effect on reducing the concentrations of VFA and fecal microbial protein. The observed improvement in peak performance is also likely due to its effect on increasing mRNA abundance of some genes involved in postabsorptive metabolism of lipids. Some things to ponder on: aside from copper, other alternatives to antibiotic growth promoters are being commonly used nowadays, such as probiotics, acidifiers, phytobiotics, and pharmacological levels of zinc. It is also a common practice to include exogenous enzymes in pig diets, such as phytase or xylanase, to increase nutrient digestibility and pig performance. However, the mechanism of action and interactions among these products have not been fully elucidated. Therefore, further research is possibly needed to focus on determining potential interactions of copper with non-nutritive feed additives to maximize the value of not only feeding supplemental copper, but also with other feed ingredients that we include in our diets. Thank you once again for your interest and for listening into this podcast. And if you're interested to learn more about other topics in swine nutrition, please feel free to visit our website at nutrition.ansai.illinois.edu.